mà giao tiếp với nhau bằng long distance, giống như là khi mà tế bào khi mà tuyến yên muốn gửi các tuyến yên muốn gửi các hormone tăng trưởng đến các cơ quan khác nhau trong cơ thể, đó nó là kích thích toàn bộ các cơ quan trong cơ thể phải phát triển, phải divide rất là nhanh, thì đó là giao tiếp long distance. Và cái trường hợp thứ hai là paragrins, là local, tức là nó chỉ giao tiếp ở trong nội khu thôi. Và với cái paragrin này thì các signaling này, các ligand này thì nó phải, thứ nhất là nó phải nhanh chóng bị tiêu hủy bởi các enzyme ngoại bào, và nó phải được nhanh chóng uptake, hoặc là phải nhanh chóng tiếp nhận bởi các tế bào hàng xóm ấy, neighboring cells. Và cái thứ ba là nó phải bị ở bên ngoài các bọn tế bào này, tức là sẽ có một cái được gọi là extracellular matrix, tức là một cái ma trận ngoại bào ấy, tức là nó sẽ có tính chất khác với các môi trường ở xa nội khu của nó ấy. Thì cái ligand này nó phải có tính chất là nó sẽ bị mắc ở đó. Và cái trường hợp synaptic là khi mà các tế bào thần kinh truyền thông tin cho nhau ấy, thì là nó short distance, đúng không? Nó tương tự với local, nó là một cái trường hợp đặc biệt của local, đó là synaptic. Nó sẽ truyền thông tin, nó gần nhưng mà nó không hề chạm vào nhau, nó không chạm vào nhau, nó chỉ là short distance thôi. Nó sẽ truyền thông tin bằng các neurons or transmitter. Các trường hợp thứ tư đấy là contact dependent, chính là không khoảng cách ạ. Đây là phụ thuộc vào contact với nhau, tức là không khoảng cách ạ. Tức là nó phải chạm vào nhau thì nó mới truyền được thông tin. Và cô lấy ví dụ ở immune cells, hoặc là hai tế bào mà đứng cạnh nhau và nó có các cầu nối, các gap junction với nhau, thì đấy là trường hợp contact dependent. Một trường hợp nữa rất đặc biệt đấy là outer-print signaling, tức là khi một tế bào mà nó tự gửi các thông tin cho chính mình. Và cái trường hợp và cái ví dụ dễ dàng nhất đối với cái outer screen signaling đó là ở cancer cell. Cancer cell nó tự tiết ra các chất để nó stimulate chính nó thúc giục chính nó phân chia nhanh hơn. Đó là trường hợp outer screens. Chúng ta nhớ là có 5 cách giao tiếp của các tế bào với nhau, phụ thuộc vào khoảng cách nhé. Oh, sorry, I have a phone call. So the first step, receptions. And for the second step, are transductions. Transductions, when how you can process this kinds of messages, right? Transductions. So for the signal transductions, a signal is a function that's conveys information about a phenomenon. So signal transductions is a process whereby one type of signal is converted to another. Here's when a mobile telephone receives a radio signal is converted into a sound signal. So when transmitting a signal is the reverse. So in B, when target cell receive information from the extracellular molecules, so they need to transmit and convert these extracellular molecules into intracellular signaling molecules. So from A, this must be transferred into B. A transfer into the B out. So for the intercellular signalings, here's intercellular signalings, it occurs within the cell surface receptors. One signaling molecules, ligands. So here, phosphorylations, I explained about the kinase. Remember CDKs, the cyclin depends kinase in the previous lectures. kinase ties of enzyme that adds a phosphate group to other molecules, and we call them the phosphorylation. It can active or deactivate other proteins. So here's one protein in the inactive state. When there is a 1P, 1-phosphate from the protein kinase, is become active form. And after they work, they will detach, they will leave the phosphate group here and become inactive states. So once signaling molecules from cell have bound to the receptor on another cells, the signaling process completes. If we are talking about the intracellular receptors, it's bind to their ligands inside the cells and directly active genes. And the answer is maybe yes. And in most cases, the answer is no, and not by a long shot. Once ligands bind to receptors, continuations of the signal in this manner is called the signal transductions. And here is one or examples of signalings by the protein phosphorylations, and the B is signalings by the GTP binding proteins. So one protein binding with the GTPs, and once when we have the GTPs binding, here is become active form, and when they are release the phosphates, they will be inactive form. Inactivate form.
So excuse me, does signal transduction only occur with the cell surface receptors? Is that right? And
so what?
I mean, does signal transduction only occur with cell surface receptors?
Cell surface receptor?
Yeah.
Yeah, they need to conduct with the cell surface receptor. And for the internals, we have another kind of the signal transduction. Okay, so keep moving forward, and I will answer your questions later. Okay. These kinds of signal transduction, the phosphorylations. So here, phosphorylations. Phosphorylation, we have an example of the MAP kinase signaling cascades. MP or MAPK, the mitogens activated protein kinase cascades. Or why is not here? Mitogens activated protein kinase. Chúng ta gọi là tiếng Việt. Cô quên mất từ này. Từ này tiếng Việt lâu không dùng. Dòng thác protein kinase hoạt hóa bằng mitogens như vậy. Các bạn có thể search và đọc qua vì cái này khá là phức tạp đối với các bạn không thuộc biên sinh. Với các bạn biên sinh thì có thể chắc là như ở high school các bạn cũng không học cái vấn đề này lắm. So, phosphorylation examples. So one, for example, epidermal growth factors. We have a growth factor here, comes or bound to the receptors. The receptor will be phosphorylated here. There's numbers of these receptor will be phosphorylated. Okay, so we have received some information from the outside, and it's activate the RAP proteins here. And they are the MAP kinase. So MKKK. After that, they will phosphorylate another type of proteins, MEKs, and it's become the MKKs only. So 1K is deleted. And then ERKs will be phosphorylated, and we have a MAP kinase. MAPKs only. And after that, the phosphorylation of the cytotoxic gets promoting growth divisions, and they will enter into the nucleus and the phosphorylations of other transcription factors here, c-mix into phosphorylated c-mix, and they will affect the transcription of the genes promoting cell growth and divisions. That's how the cells can, so they are received here, they are received here, reception here, they are transduction here. And from this, they have the response, right? This is about the mitogen activated kinase. Các bạn có thấy cái này nó phức tạp không ạ? Cái này hơi phức tạp nên là xem nào. Cô sẽ, thôi được rồi, ở bài tutor cô sẽ nghiên cứu xem bỏ cho các bạn cái phần nào nhé. So this is the map of the MAP kinase. Is actually map the MAP kinase. So is there summarizations, and is the details of the MAP kinases. MAP of the E or R casein grow in differentiations. So for these kinds of MAP kinase and the signaling inside the cells, for the student come from the life science departments, you might discover more too simple. You may discover more in the advanced cellular biologies. And for the cellular biology in the first semester, we stop here. Okay. This is a graph. Thực ra là đây mới chính là một cái biểu đồ của một cái MAP kinase. Rất nhiều các proteins nó involve vào cái này ạ. Và các bạn nhìn thấy đây, nó cứ MKK, MK, MKP, tức là cứ mỗi lần nó gần như là một chuỗi phản ứng ấy. Cứ cái protein này nó phosphorylase thì nó sẽ activate cái protein tiếp theo lại được phosphorylated, được phosphor hóa, và nó lại activate cái protein nữa, và nó tạo thành, người ta gọi là cái dòng thác đấy, signaling cascade, cái dòng thác protein hoạt hóa bằng mitogen. Tại sao gọi dòng thác ạ? Nó như thác lũ đổ, cứ thằng trước dồn thằng sau. Và đây chính là một cái graph của cái MAP kinase liên quan đến cái quá trình tăng trưởng và biệt hóa liên quan đến cái hierarchy. Phần này cô không yêu cầu các bạn học nhé. Không phải confuse với cái phần này đâu. Now, okay, it's the second case of the signal transductions. So in the first we have the phosphorylation, right? And the second case we have the second messengers. So small molecules that have to spread a signal through the cytoplasms after ligands bind to a receptors, we call the second messengers. We have an example here about the cyclic AMPs or calcium ions. So in response to the signals, enzymes called the adenines or cyclase will convert ATP into the CCAMs. So ATPs, when the ligands bind to receptors, they will activate the adenine cyclase, and they convert ATP into CCAMs. The CCAMs is going to protein kinase here. They form from inactive into the active form and causing the response. Here are the examples of the adrenalines stimulates the glycogens breakdown in the skeletal muscle cells. Adrenalines, nhớ adrenaline từ cấp 2, cấp 3 đều học rồi đúng không? Adrenalines will activate the GPCR proteins. Is actually there's receptor of adrenaline, GPCR, and they will turn on the G proteins here. They turn on the G proteins is next to them. They turn on G protein. They can activate the adenine cyclase to boot the formations of the cyclic AMP. This guy talks to the receptors and boot or activate these GS proteins. GS proteins will activate, transfer the ATP into the cyclic AMP, CAMP. These CAMPs will be they will activate the pKa here to become the active form. And the active forms of the pKas will make the inactive phosphorylase kinase, one kind of enzyme that's involved in the glycogen breakdown process, right? Inactive form to active form. And the inactive glycogen phosphorylase will be phosphorylated by the ATP here to become the active glycogen phosphorylase. So then, by this kind, this is an enzyme. Enzyme break the glycogens. So the glycogens will be break down whenever this active glycogen phosphorylase becomes active form. So do you think this response is quick or slow? How do you think about this response? It's still complicated, like, or it's okay, we have adrenaline, they met to a receptor in the cell membranes, and they will attack, they stimulate another type of protein we call the GS protein, and this GS protein will transfer the ATP into the AMPs. CMPs will go to activate the pKas. pKas will activate the phosphorylase kinase. So here's by here we have phosphorylations in this step to next. Until to this we have a phosphorylation like. And this active phosphorylase kinase will activate the glycogen phosphorylase, and the glycogen will be breakdown. Do you think this could be fast or it could be slow? Depends on what origins we choose. So it depends. If you want to, it's slow, it's slow. Or if you want, it's quick, it will be quick, right? You think it's quick or it's slow? It's not depend. It's probably slow. No. It's very, very quick. So, you know, there's numerous process happens, like I talked a lot about the process, or even in the mouse kinesis cascade, you see there's numerous of protein involved, but it's very, very quick. Like, or các bạn nhớ năm cấp 2 khi mà cái thí nghiệm mà tiêm adrenaline vào ếch hay gì đó mà cũng nhớ rõ lắm, nhưng mà cái phản ứng nó diễn ra ngay lập tức adrenaline, đúng không ạ? Đối với những người bị sốc phản vệ chẳng hạn, sẽ tiêm luôn adrenaline vào và cái quá trình nó diễn ra sẽ là ngay lập tức ạ. Tất cả quá trình này nó diễn ra chỉ một vài giây thôi ạ. Nhìn rất phức tạp nhưng nó chỉ diễn ra trong một vài giây thôi. Nó respond very very quick. Okay. So we move to the next step in the cell communications. It is response. How the cell can respond. So the response, the same signal molecules can induce different response in different targets. Different targets. So you see this the same with acetylcholines. We have acetylcholines, but when they bound to the receptors in the heart or pacemaker cells, so it decrease the rate of feelings, decrease the rate of the heart. And if they come to the salivary gland cells, it will secrete more sorry. Nước bọt là gì? Tự nhiên cô quên mất từ này. Sorry, increase the secretion of the salivary gland cells. And when they come to the skeletal muscle cells, it will make the contractions of the skeletal muscle cell. So the same signal molecules, just we have only one acetylcholine. They induce different response in different states of the cells. Okay. So in the next, every cell type displays a set of receptor protein that enable to respond to a specific set of extracellular signal molecules produced by other cells. So one cell, if A, B, C is respond to the survive, so then the cell will survive. If we have some kinds of growth hormones come to interact, they will grow and divide. Like we have a G8 come here, it will grow and divide. Or we have a several types of the ligand come to say, okay, you need to differentiate in another cell type, it will become differentiated. Or if one cell receives no information, it could be died. So if one day that you receive no information, you feel bored, but the cell, if they receive no information, so at least they receive no information from the ligand that or ensure they become survive, they will be died by apoptosis. Okay. About response, we have two types of response. So here, first, very second to minutes, lies when the extracellular molecules or signal molecules come to bind to the receptors. We have an intracellular signaling pathways, like I introduced in the previous slides. They enter the protein functions and they enter cytoplasmic machinery, so it's very, very quick. So in case they want to enter into the genetics regulations of the regulation of the genetic materials, it could be very slow from the minutes to hours. So they could be enter the protein synthesis. Ở đây phân biệt cho cô có hai cái trường hợp cái response của tế bào nhé. Nếu như mà nó không đi qua nhân, các bạn cứ nhớ là với bất kỳ trường hợp nào, nếu như cái thông tin này, cái transaction này nó không cần đi qua nhân, nó không cần tác động đến các genes ở trong nhân, thì nó là fast, từ giây đến phút thôi ạ. Nếu nó cần tác động đến các genes trong nhân, tức là có thể là tăng sự hoạt động của gene này hoặc là giảm sự hoạt động của gene này, sẽ dẫn đến là alters, thay đổi cái protein synthesis, tăng hoặc giảm của một số loại protein các thứ, thì nó cần nhiều thời gian hơn. Đấy là cả hai loại response của tế bào. So quicks in cells, change in the cell movement, secretions, or metabolism. So they not need to invalidate change in the gene expression. slows like the cells differentiations or increase the cell growth and cell divisions, and they involve in the chains of the synthesis of new proteins. There's two cases of response. So here we have some small hydrophobic hormones that can bind to intracellular receptor that can act as a transcription regulator. Here is the ligands that can across the cell membrane. So they don't need to bind to the receptor in the surface of the cell membrane, right? We have several kinds of the cortisone here. They can cross the plasma membranes and they can bind to the nuclear receptor proteins, and they can activate the receptor cortisone complex and then move to the nucleus, and they can activate the target genes, and the transcription of the target gene will be altered. So we have here the several types of hormones, steroid hormone. But do you think that's why not with the crestons? They belong to this group. So assume that if the crestons is were not normally found in the cell membranes, could it be used as a hormones or appropriate intracellular receptor? Do you think this could be? If the crestons we cannot found is not a structure components of the cell membrane, could it be one hormones like estrogens, like testosterone? Could it be? So let's think about it at home, or I think is no one's focus on my lectures right now. Okay, so but the next is the components of these or intercellular signaling pathway perform one or more crucial functions. So first, they can simply rely the signal onwards and thereby help separate it through the cells. And they can amplify the signal received, making it stronger. And they can detect the signal from one or more intercellular signaling pathway and integrate them before relaying a signal onward. Or they can distribute the signals to more than one effector protein and creating branches in the information flow the diagram. So you can see here extracellular signal molecules bind to the receptor proteins, transductions processed, and they can affect number of things inside cells. Effector proteins like enter the metabolic pathway, they will enter the metabolism. Or the cytoskeletal protein, they enter the shape of the movement, or they affect the transcription regulator, then they can enter the gene expression. So the cellular response to the signal change in the gene expressions, or they can increase or decrease the production of the protein produced by a specific genes, or they can increase or increase intercellular metabolism, or they can convert glucose to glycogen, or they can enter the cell growth. They do not normally divide unless they are stimulated by the signal from other cells. So only the cells receive the information from the outside environment. So let's grow, let's divide. So then they will divide. And the cell death also. The hypothesis is control the cell death, and the cell become stimulated, dying. If they are abnormal, they are infected with the bacteria or the virus, and during the specific part of the development. So the way the cell responds to the signal, we have 2 or 4 types of response. Alright, so now is maybe you can see videos from Amoeba about the cell communications.
A question for you. Have you ever played the game Telephone? You know, the game where someone whispers something to someone, and then that person whispers it to someone else, and then that person whispers it to someone else. And by the time you get through everyone playing, the original message is all messed up. I used to kind of dread that game. Most people seem to like it, but somehow whenever it reached me, the message was always really messed up, so I felt like I was spreading nonsense. Well, either that or it was me all along that misheard it. Anyway, the game is all about communication and how things spread. Communication is incredibly important.